Good morning, everyone. I'm Sandali Malankar, and I welcome you all for today's webinar on significance of biologicals in pharmaceutical sector, hosted by the VESCOP Alumni Association. Firstly, I would like to deliver a short overview of our committee. The Alumni Association of VS College of Pharmacy believes in being the channel of interaction between the today and tomorrow of our college. Our alumni play a crucial role in developing a greater sense of understanding of the field through various workshops which become a key asset for the future pharmacists of VESCOP. We give back to the society by educating our younger generation on topics learned only through experience, exposure, and hardships to prepare them for what lies ahead. We ensure proper learning of soft skills and professional growth. Some recent activities conducted by us include mock interviews for college students, which simulate live interview situations to train them for the corporate world, Brand Yourself activity to create awareness about personal branding, and Alumnus of the Month activity in which the alumni share their journey, give advice, and provide insight about their respective fields through indirect interaction. Various offline and online seminars by alumni speakers were arranged, which compose our Talkathon series. The topic of today's webinar is unique and a great subject of interest for pharmacy students and graduates. This is also fascinating for research projects by students and faculties. It gives a complete picture of biologicals industry scenario and various opportunities for pharma graduates. The presentation gives insights on the current trends from market research and also the upcoming trends that are shaping up the pharma industry in this subject. This webinar bridges the academic industry gap. It is a great pleasure for me to introduce our speaker, Ms. Anushka Rajesh, who is an experienced research specialist with a demonstrated history of working in the biotechnology industry. She is currently working with VIR Biotechnology, San Francisco, California, as a research associate in virology department. She is extremely skilled in laboratory, instrumentation, and computational skills, public speaking, management, leadership, and teamwork. Not only that, but she is a highly motivated researcher with a focus on patient-centric medicine and gene therapy. Creativity and positive attitude have always been her forte. She has publications in various journals, including Nature Biotechnology and BMC, and various preprints in Nature Methods, Nature Genetics, and Genome Biology journals. Her portfolio also comprises of MS in Pharmaceutical Sciences, Master's thesis in Bioinformatics, and experience as a graduate teaching assistant at University of Southern California, Summer Intern at Merck Consumer Health, and Best Student of the Year Award in VESCOP for the year 2018-19. Before we get started, I would like to appeal to all the participants to type the questions, if any, in the chat box and to keep their microphone and camera off during the session to avoid any disturbance. Questions will be addressed at the end of the session. Now let's turn the time over right away to our speaker, Ms. Anushka Rajesh. Over to you, ma'am.
Type into the chat or reach out to me later or whatever. I'm just gonna try and share my screen. Okay, I'm just having a little issue. Okay, I don't know. I'm not able to share my screen.
Yeah, Anushka, I'll just make you a co-host. 1 second.
Okay.
You can try now.
Yeah. Okay. Okay, it's just giving me an option to share from the tabs I have open on Chrome, but not a presentation for some reason.
You can try sharing your entire screen. And
I'll do that. Yeah. Okay. Can you guys see my screen now?
Yeah, yes, it is visible.
Yes.
Okay. Yeah, I cannot see any of you all. So if you all have any questions, I'll try to address it later. But first, before getting into the topic, I'd like to tell you that, although you might not know what biologics are right now. Honestly, when I did my undergrad, when I did my master's also, I didn't have too much exposure to biologics, but it is always easy to sort of explore a lot of areas in the healthcare industry and know what works best for you and what you like most. So just at this point where you are, just try and grasp as much information as you can, and then look out for yourself how you like it, and if it's something that you would probably be interested in working on later. Okay. Yeah, so before we start, like, thank you for the wonderful introduction that you guys gave. And yeah, I graduated from VESCOP in 2019, and then I went on to do my master's in USC, which is in LA in California, and I did that in pharmaceutical sciences. And I had started work in a laboratory that was working on site-directed mutagenesis and other genetic work. But unfortunately, right in the middle of that, COVID hit, and the labs actually just shut down. So because of that, I had to sort of adapt to the situation, and that is why I got my thesis in bioinformatics. And yeah, that was a great skill to develop as well. And if you guys are interested in coding or have any sort of interest in understanding how it works, it's definitely an avenue you should check out. Um, but yeah, after that, although I got my thesis in what we call a dry lab as compared to the wet lab where you actually have to go and work, it was very I was definitely very inclined towards wet lab work because I definitely wanted to work on things that are relevant to actually seeing how healthcare works. So I'm just gonna try and share some things today. Okay, so let's talk about the history of therapeutics until now. We started with um, back in back when healthcare became a concept. It all started with natural products, and we ourselves, we had Ayurveda in our country, and there were a lot of remedies that came from plants or animal products, and that is where healthcare actually started. Slowly it developed onto small molecule drugs, as now we know as the chemical drugs that we use, the synthetic drugs. And the first of this was chloral hydrate, which was a sedative hypnotic. Then, as time passed, many of the small molecule drugs that were developed were sort of modifications of structures of natural products that were known to be good remedies for certain conditions. And now, where we are at this moment, there's a lot of importance towards biologics, especially now that the pandemic hit very recently. It's become all the more important to try and understand how we can use biologics to develop better remedies, which are probably more effective, or at least definitely comparable if efficacy to the small molecule drugs. Okay, so what are biologics, right? We're keeping on talking about biologics, and it's important to understand what they are. Biologics are basically molecules that are very large, they are very large molecule compounds. Here you can see they are definitely more than 20,000 atoms as compared to a small molecule, for example, aspirin, which is probably just 21 atoms. Biologics are usually derived from living cells or any components of a living cell. And the first known biologic product was the diphtheria antitoxin that was developed derived from the horse serum by Roche in Germany. Okay, so there are three main mechanisms by which biologics can act as a therapy. The first is that they turn something on and they act as an agonist for a certain pathway in the body. For example, they can act on enzymes or ligands that can be, but the disadvantage is that they can be cleaved by proteases in this case, if they are proteins, and that makes their half life very short inside the system. The second mechanism that they can act by is by turning something off in the pathway, basically acting as an antagonist to a certain cellular pathway. Um, for example, the membrane protein, extracellular FC fused protein. I wouldn't go too much in detail, but this is just an example of you can either turn something on in a pathway and act as an agonist, or turn something off, knock it down, and act as an antagonist. The third mechanism is by completely removing something from the system, and this is target cell killing. So, in this case, for example, antibodies that engage the immune system to kill or antibodies that engage secreted proteins to remove something from the cell. The pros are that you have a long half life, the dosing frequency is better, in terms, so it's lesser, and that sort of also enhances the patient to follow the dosage regime. And antibodies are definitely very, very specific. The cons of using antibodies is that sometimes they're too specific and species cross reactivity is limited. So if you have an antibody that you've got from a certain species, it may not act the same way in human beings, and that is a disadvantage. Also, the dosage usually is pretty high. Okay, so according to the section 351 of the Public Health Service Act by the FDA, this is the definition of a biologic. It says the term biological product means a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, allergenic product, protein, except any chemically synthesized polypeptide, or analogous product, or arsphenamine or derivative of arsphenamine, applicable to the prevention, treatment, or cure of a disease or condition of human beings. I know this sounds very complicated. A lot of terms are being thrown in here, but basically, what it again means is that you're deriving something from a living cell or from a component of a living cell, and you're modifying it in a way that makes it helpful as a therapeutic product for human beings, for treating many, like a broad range of diseases. So the sources for biological products are humans, human cells, human tissues. It can be any of these. The blood components, even the RBCs, WBCs, all of that do count as biologic products. The next is mammalian cell culture, where you sort of get a cell line from a species, be it human or be it any other animal, and then you culture those cells. You add certain substances to it to enhance certain pathways in the cell, and then derive products from that. You can also get it from mice, from bacteria, and from transgenics. Okay. Like I said, the types of biological products are blood derivatives, proteins, definitely antibodies, vaccines, for example, now the COVID vaccine that we've all been taking is also a biological product. Then you have allergenic extracts, you have cell and gene therapy products, and human tissues. And these can be used specifically for different conditions, and different biological products work better for different conditions. Okay, so the main differences between small and large molecule drugs are that small molecule drugs, definitely because they have a very small chemical structure, their molecular weight is very low, while biologics are very large molecule, more than 28,000 atoms, and they have a very high molecular weight. The next is small molecule drugs undergo organic or chemical synthesis, while biologics you can derive from genetic engineering or by tissue culture. So small molecule drugs, the structure is very well known, and it is a fixed structure because you cannot really change the structure of a chemical compound and expect it to work the same way. Sorry. But for biologics, it is more complex, and sometimes the structures are partially unknown, which sort of makes it also difficult to try and manufacture biologic products the same way as something that has already been established. For small molecule drugs, it's less. The weight is less than 1 kilodalton, and for biologics, it's more than 10 kilodalton. Small molecule drugs usually, in comparison to biologics, have a lower immunogenicity because definitely they'll still be treated as a foreign substance in the body, but they won't um, express as much of a immunogenic response as a biologic product would. Lastly, small molecule drugs are easy to characterize, be it the structure or other physical and chemical aspects, while it is more difficult and complicated for a biologic drug. Okay, The advantages definitely is that the onset of action for biologic drugs is very quick because the mode of administration is usually intravenous or subcutaneous, usually. They're relatively safe to use. I know I just said that the immunogenicity might be higher, but what I mean by safe to use here is that you can definitely inject a biological drug into a human being, and there might be immunogenic responses, but it's always safer than a synthetic or a chemical drug. Okay. The drawbacks are, like I said, the risk of immunogenicity because the body would just consider it to be a foreign substance and produce an immunogenic response based on that. Secondly, the complexity of the manufacturing process because biologics, part of their structures are sometimes unknown, which doesn't make them, you cannot have a product sheet which defines all of their characteristics completely. And thus, even replicating the process sometimes is a bit more challenging. Um, Another thing is that you need to adjust the dose according to from individual to individual because someone might react differently to a biological drug and someone might react in probably another way, and then you have to figure that out. So it's a little more complicated for even the physicians to um, decide what kind of dose adjustment needs to be done based on the conditions of the individual and any pre-existing conditions if there are. And also, care needs to be taken while co-administrating biologic drugs with synthetic drugs, as is true for synthetic drugs also. Okay, I just want to broadly introduce the concept of biosimilars here as well. Biosimilars are basically biopharmaceutical drugs designed to have active properties that are similar to previously licensed product, which is a biologic, and that has been approved by the FDA. So here, previously licensed product basically means a biologic. It is similar to how we have generic drugs for the chemical drugs. And the advantage of biosimilars is that if you figure out what the manufacturing process would be like, it is good to have a biosimilar because there's no significant difference in terms of safety and efficacy from the original biologic. So that yeah, this is actually the end of my presentation, but yeah, from my industry experience, I want to talk more on that because I'm, like I said, although I didn't have too much of lab experience in biologicals before, now the industry, the way it is transitioning, is definitely giving a lot more importance to biologics. And I have just had a lot of experience. Most of my work right now actually revolves around cell culture and tissue culture, and doing many assays on those cells that I culture. And it's definitely, if you, any of you are planning to, go in for higher studies at some point, definitely biologics is an avenue you should check out because it's where we are now. The pandemic has definitely shown us the importance of it, and we know that it is the future of the pharmaceutical industry. So if you ever get a chance to explore this, you should definitely take it up. And yeah, I think I can take questions now if you guys want to ask anything, or if you want me to talk about more of what my work actually entails, I'll be happy to do that as well. Any questions? Was it too quick, or do you want me to go over something better? Okay, so Tejasvi asked how biologics are different from biosimilars. So biologic is the original drug that has been developed is called a biologic. And once it's been approved by the FDA and someone tries to manufacture a similar product with similar safety, similar efficacy, but not the same, it's called a biosimilar. Does that answer your question? When it comes to large molecules, is it feasible to do isolation and characterization at lab level? You definitely, it's like I said, it's definitely more complicated to characterize biologicals, but you definitely have specialized lab equipment that you can use. Personally, I don't work too much with characterization, so I don't know if I'm the right person to answer this, but I can definitely, if you send me an email, I can get back to you with a proper answer. If you're looking, I can ask around, and definitely get back to you. Are biologics better than biosimilars? No, because like I said, the safety and efficacy is pretty much comparable, and they are, like I said, they're like the generic version of, say, a branded drug. If you want to look at it in that analogy, you have a branded drug and then you have a generic version of it. It's essentially the same in terms of safety and efficacy. So biologics and biosimilars are pretty much the same. Career prospect in biologics, I'd say the industry in general is definitely moving more towards biologic products. Small molecules definitely play a large part still in treating most of the diseases and illnesses, but biologic products are definitely on the rise, especially for, say, viral infections. Like now we've had COVID, and we need vaccines for that. We need monoclonal antibodies are so much on the rise for a lot of, even for rheumatoid arthritis or many other indications. So the career prospect is definitely, there's a lot of prospect, and um, yeah, it's definitely a good skill to develop if you want to explore that area. Okay, I got a question on bioinformatics as well. So, bioinformatics basically, I'd say there are two types of things that you can do under bioinformatics. One is analyzing data that's publicly available, genomic data that's publicly available, or one is benchmarking. So first I'll talk about and the analysis part. Basically, you have a lot of these. There are these things called repositories on the internet, and repositories basically you can picture them as storage bins for a lot of data. So when researchers actually collect data for a variety of illnesses, say sepsis or tuberculosis, any of those data, they usually once they publish their data in a paper, they can go ahead and also put it in this public repositories, which is accessible to the common man. And you can use bioinformatic tools for studying the DNA or RNA alignment. You can do many other, you can find out the frequency of a particular condition. You can do a lot of things like that using bioinformatic tools. That is the analysis part. And then the other part would be benchmarking, which is basically you have certain a variety of tools that perform the same function. For example, RNA alignment, if you see, there are probably 200 tools that are available. But so benchmarking, basically, you're trying to compare these various tools with one another and see if all of them are optimum and are giving you accurate results or not. Okay. The next question. What type of projects can be taken up in biologics at farm level? Okay, thanks for the question, Harsha ma'am. So the question was, what type of projects can be taken up on biologics at an inform level, and what are the minimum facilities or instruments that are needed? Is this feasible by pharmaceutics faculty, or you really need a biotech specialization? So I'd say that the facilities and instruments that you would need, the basics would be to have probably a tissue culture or cell culture hood, and the facilities that go along with it, like a vacuum pump to aspirate media that contain a pathogen or stuff like that. And it is feasible by people who are in pharmaceutics as well, because like I said, even I had no experience. But if you have someone to guide you through it, you can definitely, it's something you can pick up, because A lot of it is repetitive work. Once you develop that skill, you can go ahead and do a lot of tissue culturing and cell culturing, and design your assays based on these and get your results. Um, okay, I have a question about my experience studying at USC and the scope for pharma graduates. Um, Personally, I would definitely encourage people to go in for higher studies, because be it MPharm or MS, it doesn't matter. The more you study, the more actual experience you get hands on working in a lab. It's uncomparable, and you get to learn a lot more, and you know what kind of challenges are there. So higher studies, definitely I would encourage people, but it's your choice, of course. So about USC specifically, there is, like I said, a lot of scope, because even the research laboratories that I was trying to do my master's thesis in, most of them are actually working on biologics, or if they're working on the pharmaceutics side, it's a lot of nanotechnology based, or modifying the chemical structure, research like that. But most of them are working with genetics and biotech products. So yeah, that gives a lot of scope, because the industry uh, is giving a lot of importance to that at the moment. Can you suggest a few public repositories from where we can get the info? Yeah, so the main public repository I use is called NCBI Geo, like it's just NCBI GEO. You can just Google it, and the first search result that you get is where you can get the public data from. But for that, you'll need an accession number from. So, usually, if you've seen articles, there is also supplementary information in papers that are published, and there they might give you an accession number for a public repository. So all you need to do is copy that accession number, go to whichever repository they have pasted their data in, and using the accession number, you can basically just get all of their data. You can see the paper again. You can get individual sample data as well. So it's very helpful. Okay. Yeah, I think those are the questions. If anyone has any other questions, or you want to know anything that I didn't talk about today, but you just want to ask in general, is also okay.
Ma'am, actually, we have a few more questions. First question is, when it comes to
Okay.
biologics, what kind of research projects can be taken up by pharmacy students?
Um, you mean while you're doing your B farm at that level?
Hello, am I audible?
Yeah. So at the B farm level,
Yes,
I'd
ma'am,
say
yes.
maybe for a basic thing, you can just start with, projects based on whatever we try to do in the bio. Okay. Yeah, like I was saying, at the BPharm level, to start off, basically to just get some experience initially, you can probably design, take your faculty help and try to design a project which is extrapolated from what we kind of do in the biotech lab. Because I know we work with a lot of bacterial work in the biotech lab. We do a lot of plate streaking and all of that. Um, but yeah, I mean, you can a basic project that I can think of is maybe just analyzing effect of, say, a natural product on a certain type of bacteria or something like that. That might be helpful to start off with. Yes.
Hi Anushka, this is Priya.
Hello, ma'am. So nice to see you.
Same here, and your talk was really very
Okay, ma'am.
informative. And
The
as
last
rightly mentioned by
question
you, there is a
is
huge career there in biologics, and you have very nicely explained. Please go ahead.
Yes, ma'am. Continue,
The
please.
last
Sandali, please hold on.
yes, ma'am.
No, what Anushka, what I felt is that I have one slide to share which emphasizes the topic that you have chosen today. So just give me a moment to share the slide.
Sure.
So, is this slide visible to all of you all?
Yeah.
Yes.
Yes, ma'am.
So you can see the spread of these biologicals, which now are available as bio. These are all branded drugs. So as she rightly said, they are not biosimilars. They are all branded drugs. So you can see these are all very big brands. And if you could see the sales turnover, it is in US billion US dollar. And you can see one molecule has a turnover of something like 9.2 billion US dollars. And just see Avastin, then Humira from Abbott. So that is for rheumatoid arthritis. Roche Avastin, it is a monoclonal antibody for cancer. Diabetes, you all know Aventis has Lantus, and Novo Nordisk has Novolong. These are all human insulin. So all these molecules, they are coming from the cell or live cell origin, as Anushka mentioned. And if you see, there are more than 20 such brands, and each costing more than 1 billion US dollar. So if we talk about 9.2 billion US dollar, that's more than the size of the Indian pharma industry. So you can see you guys have got a great career there in this field, and you must definitely explore this particular field very actively the way Anushka is doing. And if you want to go abroad, definitely you can try for biologics, biosimilar area. And even in India, if there is opportunity to do doctorate, postdoctorate, you must uh, think this as one of the important alternatives. I felt I can add to what she said because it is really very important topic, and I must thank our alumni association in charge, Anita madam and Rashmi madam, to invite Anushka. Anushka is a very brilliant student of VESCOP, and she has been performing very consistently right from first year till final year. Not just that she was good at academics, she has shown all round performance. She was our best student. And I guess she was a best student at pharmacy colleges level also, Anushka. I really
Mm,
don't remember, but yes,
no.
I just know her track record, which is absolutely brilliant. So
Thank you,
it
ma'am.
was really a very thoughtful decision of our alumni association to invite you, and I felt I must catch up with this session that Anushka is coming. So
Uh,
over
Makes
to
me
you,
feel so
Anita madam.
nice.
Anita madam, sorry I interrupted. Some questionnaires were going on. You carry on. Continue.
No, no problem, ma'am. It was a very important value addition because the purpose of having this session is that so that the students are aware of the different avenues. So it really added to what Anushka had discussed. So Sandali, you were taking few questions, so you can continue.
Yes, ma'am. Thank you so much, Principal Ma'am, for sharing some additional information on this topic. We just have a last question. Is MPharm in biotechnology the right choice to go for career in biologics?
Yeah, I think it's definitely, like I said, it's a very interdisciplinary sort of field that the healthcare industry is. So no matter what you do, your masters or be it MPharm or MS, even if you say do it in like personally, even I did mine in pharmaceutical sciences, and that was just a title, but it's it's so interdisciplinary in a sense that if you have the basic knowledge you can definitely explore a lot more. So yeah, if you go for a MPharm in biotechnology, it's definitely going to give you a very strong basis for developing the skill set that you need for a biologics career.
Okay, that's all the questions we had. Thank you so much, Anushka
Sure.
ma'am.
And I would just like to add that even if you're not able to get hands-on experience right now at the B Farm level, it's definitely very important to keep in touch with what the industry is shifting towards, and it's very important to keep reading published articles and papers that have been coming up. And now during COVID, there have been so many publications. You can get so much information, and it's very important to keep yourself updated and don't just stick to the academics. I know it gives you a very strong basis what you're learning in BPharm, but you have to sort of keep reading and keep learning, keep getting as much information as you can through these articles, and that's extremely important.
Right. Thank you. It was really an insightful session, and I'm sure all the participants gained vast knowledge about biologics.
I'm so glad I could be a part of this, and I thank you for inviting me again. And if you have any other questions, I think Anita ma'am has my email ID and phone number as well. So please feel free to reach out anytime about anything, and I'll be happy to help in whatever way I can.
Yeah, sure. I'll be making sure that all the participants have your connect. So if at all they have any specific questions, they can connect to you one-on-one. And
Yeah.
I would really like to thank Anushka, considering the difference in the time zone. Principal ma'am, when I approached her first time, she was not even hesitant even for a second to say yes for this session, and she was like, I'll come from my work and I'll be there for the session. So that kind of an attitude and having that kind of compassion for your alma mater, that is something really commendable. So thanks a lot, Anushka.
Not at all. Actually, it is my privilege. Like I said, it's I'm still so connected to VESCOP, and it's given me so much. It's definitely my pleasure to actually come and speak to all of you.
Anushka, just one last request.
Yes, ma'am.
If there is any opportunity or any possibility, sooner or later, whenever you have convenient time and resources, you help us in setting up a good cell culture lab at VISCOP,
Sure, I would be happy to help. Definitely, ma'am.
because that's one of the agendas that VISCOP has, and
That's
somehow
really
this
nice.
decision is not getting implemented. So someone like you can definitely take a lead role, and we all would work under your guidance. But we want to set up one lab.
No, ma'am, I'm not any expert, but I will definitely be very happy to be a part of this for sure. And I think it will be very helpful for the students also to get that experience already for sure. Okay. Thank you so much, everyone. And yeah, please don't hesitate to reach out if you have any questions regarding higher studies or any bioinformatics, biologics, any of it. I'll be happy to help.
Anushka, can you post your email ID if you don't
Yeah,
mind?
yeah. Yeah, that's my email ID. I can actually also give my number if you guys need. I'm actually quicker replying to WhatsApp messages than emails sometimes, so whatever works. Okay.
Anushka.
Yeah, Anushka Hirlekar ma'am is here.
Hello, ma'am. How are you doing?
I'm very much fine, thank you. I was in the practical and I was attending from there. Yeah, so nice to see you. Thank you so
Same,
much.
same here, ma'am. Thank you.
Yeah.
Okay. Thanks, everyone. Bye.
Thank you, Anushka, for a wonderful session.
Yes.
And thank you, Principal ma'am. Thank you to all the faculty.
Anita, madam, have you taken a screenshot? Anita,
Yes,
madam?
yes.
Yeah,
Throughout
already.
the yes, ma'am, we have taken.
Yeah, sure.
Yeah.
Okay, bye. Thank you
Bye.
so much,
Thank you.
Anushka. Stay in touch.
Thank you. Bye.
Yes. Bye.
Bye.
All the participants are requested to fill the feedback form link in the chat box. We may now conclude the session with a vote of thanks by Ms. Tejasvi Dubey.
Okay, good morning to everyone present here. I'm Ms. Tejasvi Tube, and it gives me an immense pleasure to deliver the vote of thanks for this event. First of all, I would like to extend my gratitude to our principal madam, Dr. Supriya Shidai ma'am, for a constant support and motivation which enables us to conduct these activities. We also thank you for adding your piece of wisdom to this webinar. I would like to thank our speaker, Ms. Anushka Rajesh, for her precious time and sharing her experience with all of us. We truly indebted for her presence today. I would like to thank our teacher coordinators, Mrs. Dr. Anita Hire ma'am and Mrs. Rashmi Vani ma'am, and all our fellow committee members for their constant support and sheer confidence, which played a crucial role in making this event a success. Special thanks to our participants, who I'm sure will be make the best out of what they have learned here. Thank you all for your patience and for maintaining the decorum throughout the event. Recorded video of this session would be available on the VESCOP YouTube channel shortly. And stay tuned and please follow our social media handles for the constant updates of any future events. Thank you and have a great day.